Okay, so I'm going to talk to you guys about enzymes and proteins. One of the things that you have to know is that proteins are made up of chains of amino acids. So amino acids are the monomers for proteins. And enzymes are proteins. But don't forget, all enzymes are proteins, but not all proteins are enzymes. And proteins are used for different functions. So here we have the primary structure of an enzyme, which is simply a amino acid chain. And don't forget, if you remember, we have an amino group, which is the N with the H, and a carboxyl group, which is here. So anytime you see those two, you'll know that it's an amino acid. So this is an amino acid chain. And then you have to understand that it's not just a long chain of amino acids stuck together. We actually get the chains actually interact with one another through hydrogen bonding, and we know those are weak hydrogen bonds, but if there's a hydrogen here and this molecule is a little bit polar, then the positive end of one will react with the negative end of another, and we'll get some hydrogen bonding. So this is what it looks like if it were a chain, and then those chains, those parts of the chains actually interact with one another, and we get this type of structure. And then we get bonds between those the parts of the molecule, because they're reacting with one another. And then finally we get folded up structure where other compounds are actually associated with the protein. So you have to understand that proteins are pretty complex structures, okay? And you also have to remember that we use very simplified drawing. So this would be like that folded up ordinary structure of protein. So this is a very simplified way of looking at it. So we have to understand enzymes as things that help chemical reactions occur. And the chemicals in this case, we're going to call the substrate, and those are the reactants in a chemical reaction. So the reactants are things that you start off with, reactants, and we call those a substrate because those are the part that are going to join the active site. And if you look, the reactants have to be pretty specific to the enzyme. So if this enzyme had a different shape, like say something like this, then these reactants wouldn't be able to join the enzyme. They would not be specific to that enzyme. So every enzyme is specific to its substrate. And it actually helps the reaction proceed. So the substrate goes to the enzyme, and we call this an enzyme substrate complex because the complex is those things joined together. So this is a complex, sort of like an apartment complex is a whole bunch of apartments together, so we call it a complex. This is a enzyme and substrate together, we call it a complex. And then if you notice that I'm showing a chemical change here, because a chemical change, there's usually a change in color, but this is a really good diagram that shows you that the color changes. So there's something happening here when the enzyme is associated with the substrate. And what is happening? A chemical reaction, which actually breaks the bonds between these chemicals to make two new chemicals, which are the products. So we start off with the reactants, the enzyme does its job and makes something new, a product. And that's how a chemical reaction proceeds. We have reactants, and then we have products. Okay, and one other way that enzymes can work is they actually can put things together. So here we took things apart, and we can actually proceed so that we have something. Oh, that's not a very good diagram. I can put it over here. So let's say we start off with two separate things, and an enzyme comes along that is specific for them. Those actually reactants can become part of the enzyme complex, and then we end up with this type of situation. And this is the the romance sitting on a couch scenario that I talked about in class. And we put the nice romantic movie on, and then the couch ends up making a connection between the reactants or the substrates, and then you actually end up with a product that is a combination of the two. So we went from 2 into 1, so that's a chemical reaction. We took 2 reactants and made 1 product. And here's another reaction where we took 1 reactant and made 2 products. So those are both chemical reactions that are facilitated by enzymes. Okay, so let's go on to the last thing that we need to understand about catalyzing enzyme catalyzed reaction. So catalyzed is something that enzymes do. They catalyze reactions, and that's a really fancy science word for help. They're helpers. So enzymes are helpers. They help reactions proceed so that they're catalyzed. So if you look here on this curve, this shows an uncatalyzed reaction with the dots. So if an enzyme wasn't present, this is how much energy would be required by the reactants to absorb in order to make the product. So you would have to put a lot of energy in it. And a lot of times our bodies don't have either that much time or that much energy to create products. So we have these great compounds in our bodies called enzymes that help things happen faster. So you think about our experiment that we did the other day where we took our milk and our vinegar and we actually helped to make curds and whey much faster because we catalyzed the reaction. And don't forget, if we had left the milk in the fridge for 4 or 5 weeks, we would have gotten the same product, but geez, it would have taken a lot of energy and a lot of time to get that. Whereas here, if you take your reactants and put a catalyst in, which is an enzyme, it actually lowers the amount of energy and you get your products much more quickly with much less energy.